Hello students, we're going to start a whole new subject today, a whole new topic today in biochemistry. This will be lipids part 1 of lipids. And the first thing I want to do is give you an overview of lipids and what molecules, I'll list for you what molecules are usually discussed in a complete context of lipids. All of these are not lipids, but you have to discuss these molecules to understand what lipids are and what lipids do. So let us begin. A little definition of what a lipid is would be in order. Okay. Let's see here. Don't have the right thing. Here we go. Lipids. Lipids are they're one of the 4 macromolecules, but of the 4 macromolecules, proteins, the 4 macromolecules are proteins, nucleic acids, carbohydrates, and lipids. Pencil, PNCL. And of these, the lipids are the most water insoluble. They have very low polarity, and so they have low solubility in water. So these are going to be your lipids. Your lipids are found in lots of places. We use lipids primarily to store energy, like fats. Lipids are fats, and so we use our lipids to store energy. I just give you 3 uses. Lipids are found in the membranes, in cell membranes. And also, lipids can serve as signal molecules. Like remember IP3 and phosphatidylinositol 2-phosphate, PIP2. So they can serve as signal molecules. Lots of these: arachidonic acid, prostaglandins, trying to put signal transduction. They are involved in signal transduction, but they serve as signal molecules. Okay, and those are some of the uses of some of the functions of lipids. Now let's talk about what molecules need to be discussed in the context of lipids. Let's put lipid or lipid-associated molecules. Well, let's just put lipid-associated molecules. Some of these are lipids, so be aware of that. First, fatty acids. We'll discuss what fatty acids are very soon. Fatty acids, things that have lots of, they have a carboxylic acid attached to a very long alkyl group. Those are your fatty acids. Glycerol. Remember glycerol, 3 carbons, polyhydroxy. Glycerol is really a sugar alcohol, and so it's reduced glyceraldehyde. So we have glycerol, and then we have glycerols can join with the fatty acids to make triacylglycerols. We can have diacylglycerols, 2 fatty acids attached to glycerol. We can have triacylglycerols, 3 fatty acids attached to glycerol. That's how energy is stored in our body. If you have 2 fatty acids and add a phosphate, we can make phospholipids. So we can have phospholipids. other molecules that we'll discuss later: sphingolipids, waxes. Waxes are considered lipids. And then some other molecules that we'll discuss: terpenes, and lastly, steroids. And what we'll do as we go through these lectures, I will continue to come back to this slide to show you where we are. And so the first thing that we're going to discuss will be the fatty acids, and we'll discuss those on the next clip. These are the lipids. The first molecule that we will discuss associated with lipids will be the fatty acid. So I'm going to draw for you a very simple fatty acid A. So it'll be a 12 carbon fatty acid. Okay. Let's see here. Let's move this out of the way. Okay, I'm going to draw a fatty acid here. Now, fatty acids are long hydrocarbon chains with a carboxylic acid at the end. And so let's draw a 12 carbon fatty acid. And so let's put our carboxylic acid group here. Okay. And then that's going to be carbon. That'll be carbon 1. And so this would be carbon 2 here. Carbon 3, carbon 4, carbon 5, carbon 6, carbon 7, carbon 8, carbon 9, carbon 10, carbon 11, and carbon 12. Now I'm going to put my bonds between these to make my long hydrocarbon. Okay, that's my 12 carbon hydrocarbon. I'm going to number the carbons here. And so the carboxylic acid group has the highest priority. So this is carbon 1, carbon 2, carbon 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So there are two components of a fatty acid. Let's do this one first. This is our long hydrocarbon chain. Hydrocarbon chain. And we call this the tail of the fatty acid. And here we have our terminal carboxylic acid. And so this is the anatomy, if you will, the anatomy of a fatty acid. Okay, and so all of this is called a fatty acid. Fatty acid. And now, whenever you have a fatty acid attached to something, for example, if you got rid of this H here and the O were attached to something, you will call that group. It's like having, like CH4, that's methane. But if you attach methane to something, we call that group a methyl group. That's a methyl group. And so the same thing for the fatty acids. if you have your long chain OH, this is the fatty acid. But if you have the fatty acid attached to something, you call that group a fatty acyl group, or simply an acyl group. Okay, and so we can have these acyl groups attached to lots of other things, many other things. Well, one thing in particular we're going to attach this acyl group to make our lipids proper. Let's go ahead and name this. Let's name this fatty acid while we're here. Let us name this fatty acid. Since this fatty acid has 12 carbons, I don't really have anywhere to write this, but I'll put it right up top. Since it has 12 carbons, we can call this deca. Deca means 10. So if this fatty acid had 10 carbons, we would call it decanoic acid. That's if it had 10 carbons. But it has 10 plus 2, so we have to add 2. It has 12, and so we call it decanoic, because do means 2, deca means 10, so it's 210 or 12 carbons. So this is dodecanoic acid. Also, dodecanoic acid, that's the systematic name. It has a common name, and the common name is lauric acid. Lauric acid. And we're going to go through all of these all of the names of the common fatty acids systematically at some point. But first I want to go over some of the anatomy with you, some of the terminology of fatty acids with you. And so here we have our first fatty acid, dodecanoic acid, also known as lauric acid. And so just a review. Fatty acids have a terminal carboxylic acid group, and they have a long hydrocarbon chain. This one has the entire fatty acid is 12 carbons, so the hydrocarbon chain has 11 carbons. So this is a fatty acid, and the functional group of a fatty acid, which attaches to something else, is called an acyl group. So we could call this group here a lauryl group, or a dodecanoyl group specifically, but we'll just call it an acyl group. And here we have our first fatty acid, lauric acid. Hope you understand it. This next clip is going to be very similar to the previous one. On the previous clip, we looked at a 12 carbon fatty acid, and on this one, we're going to look at a 16 carbon fatty acid. I really wouldn't show you this one. The only reason I'm showing you the 16 carbon fatty acids is because I want to compare this one to the next clip. And so let's draw this one, 16 carbon fatty acid. Okay, let's first draw our terminal carboxyl. So that's 1, okay, CH2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, and 16. So we have 16 carbons here. Now let's connect them with our bonds. Okay, and there's a 16 carbon fatty acid. can name this fatty acid. I'll give you 3 ways to name it. The first way we can name it is we can give it a simple number name. And the simple number name for this fatty acid would be 16. And there are no double bonds, so this would be 16, 0. So this would be fatty acid 16 colon 0. That would be a name. Then we have our systematic name. And the systematic name for this one, well, if there were 10 carbons, it would be decanoic acid, if there were 10 carbons. But there are more than 10. There are 16 carbons. And so this is going to be hexadecanoic acid. If we numbered our carbons, that would be 1, 3, 5, 7, 9, 11, 13, 14, 15, 16 carbons. So this would be hexadecanoic acid. That's the systematic name. And finally, we can give it a common name. There's a common name for hexanoic acid, and we call this, since this is very abundant in, how should I say, palm trees. This acid is, or this fat is, and so we call it palmitic acid. Palmitic acid, and it's called palmitic acid because, let's see if I know how to draw this. Okay, let's see if I can draw some nice fronds here. I don't know. Maybe that's what a palm tree looks like. I don't know. There's a palm tree, and this lipid, or this fatty acid, palmitic acid, is very abundant in palm trees, hence the name. And so this is another example of a fatty acid. Anything else I want to say about this one? Oh, if you were to look at this fatty acid here, notice that every carbon in the long hydrocarbon portion, every carbon is filled with hydrogens. You can't add any more hydrogens because this carbon has all 4 of its bonds. Carbon 2, 3, 4, 5, 6, all of them have all 4 of their bonds. And so they're filled with hydrogens. And because they're filled with hydrogens, we say that they're saturated with hydrogens. Saturated with hydrogens. And so we can say that all of the carbons are saturated with hydrogens. All of the carbons in the hydrocarbon region. And so, for that reason, we say that this fatty acid is a saturated fatty acid. If it had a double bond in it, it would not be saturated. So any hydrocarbons that have any fatty acids in which the hydrocarbon portion has a double bond is said to be an unsaturated fatty acid. And we'll look at an example of this on the next slide. On this clip, we're going to create another 16 carbon fatty acid, but in this one we're going to put a double bond in it. And so let's draw a 16 carbon fatty acid with a double bond, and we'll put the double bond on carbon number 9. Seems to be a very popular one. So here we go. I'm going to put my So that's carbon 1, 2, 3, 4, 5, 6, 7, 8, 9. Well, I'll do something else. 9, 10, 11, 12, 13, 14, 15. Well, something is wrong because 15 can't be down. So that's let me number these. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. Yeah, 16 would have to be down. Okay, and so here's our 16 carbon fatty acid. Let's put our bonds here. Okay, we're going to do one thing different here. On this one, carbon number 9, we're going to have a double bond. Okay, I'm going to put a double bond. So we can't have all of these, have too many H's here. And now the way it's drawn, okay, there's a double bond. Okay, I'm going to put this H here is going to be here, and this H is going to be here. Okay. Now on this double bond, if this double bond, you can see here, this H and this C here, they're on the same side of the double bond. But the two C's are on opposite sides of the double bond. And so if I drew just this double bond out like this, I'm just going to draw this double bond here. So the way I have it drawn, so this H is going here, this H is going down, and the rest of the hydrocarbon chain is on the opposite side. And so this double bond here, as it's drawn, have the two alkyl groups, or the two R groups, across from one another. So they're across from one another. And so we call this type of double bond a trans double bond. And in a trans double bond, it leaves the hydrocarbon chain straight. And so the hydrocarbon chain is still straight. Now, this is not the type of double bond that's found in us, in human beings, in mammals. A few bacteria can do this, but no higher organisms have trans double bonds. So a trans double bond is when the two hydrocarbons are on opposite sides of the double bond. The opposite of that is a cis double bond. And a cis double bond looks like this. Okay, we have the hydrocarbons, the two hydrocarbons are on the same side. So with this one, this leaves with the trans double bond, that leaves the hydrocarbon chain pretty straight. But with a cis double bond, the hydrocarbon chain has got a bend, or it's going to have a bend or a kink in it. And so the one that we've drawn here, this is still hexadecanoic. Well, it's not hexadecanoic. Let's give it the number name first. The simple number name. I don't have any room, so I'm just going to put simple. Just put simp. It's going to be 16,9. So it's a double. I'm sorry, not 16,9. It's going to be 16,1. So it's a 16 carbon fatty acid with one double bond. The complex number name would be 16,1-delta-9. And so 16 means it's 16 carbons. 1, there's one double bond. And the delta 9, the Greek letter delta is like our D. So it's like D9. The double bond is at carbon 9. There's also a systematic name. I'll just put cis. And it's not hexa. If there are no double bond, it would be hexadecanoic acid. But it does have a double bond, so it's no longer an alkane anoic. It's an alkene, so it's an enoic. So it's hexadecanoic acid. So this is hexadecanoic acid, and it's trans. And so trans fatty acids are not found in living organisms. So you can completely forget about this one. You won't find, so this is a trans double bond here. You won't find this. This is the one, cis. It's the cis double bond that's found in organisms, in living organisms, except for a few bacteria. And so we're going to stop here with this fatty acid that's not found anywhere. We're going to stop with this one. And we're going to look at the correct one, the cis fatty acid. I didn't give you the common name. The common name for this one is going to be, so the common name for the 16 with no double bonds was palmitic acid. And so with a double bond, we call this palmitoleic. This is palmitoleic acid. Okay? But this is trans palmitoleic acid. And remember, this one is not found in living organisms. So we're going to stop here with this one, with trans palmitoleic acid. We're going to, on the next clip, we're going to look at the correct fatty acid. So of course, this is now a double bond. We say that this one, I keep adding things because I keep remembering things. Because there's a double bond, this is saturated. This is a saturated fatty acid. Excuse me, an unsaturated fatty acid. Because those two carbons, carbons 9 and 10, aren't filled with hydrogens. They only have one hydrogen each. They can absorb more hydrogens. So this is all I'm going to say about this incorrect trans fatty acid. I'm going to stop there. On this clip, we're going to draw a fatty acid with a double bond in the correct orientation, not trans, not a cross, but cis. Let's draw this one. And it's going to be a 16 carbon one again. And so let's get right to it. Okay. Let's start here with our, well, I guess it doesn't matter. Okay, and that's 1, 2, 3, 4, 5, 6, 7, 8. 1, 2, 3, 4, 5, 6, 7, 8. Okay, and this is going to be 9. 10. 14, 15, 16. Okay, there we are. Now let's put our bonds. Okay, these are going to be our bonds here. Okay, of course, I'm going to put these bonds here. Okay, now between 9 and 10, I'm going to just do a little numbering. 1, 5, this is going to be 9 here. This is 9 and 10, 12, 14, 16. Okay, let's put our double bond here. There's our double bond. And now we're going to put our hydrogens here. And the thing that you want to see here is that the two R groups, they're on the same side of the double bond. And when you have your two R's on the same side, this type of double bond is called a cis double bond because they're on the same side. Cis. And when you have a cis double bond, that puts a bend in the hydrocarbon chain. And so you can see here that the hydrocarbon chain has a bend in it. And that bend is also called a kink. And that kink is very important physiologically. If you have straight fatty acids can pack together very tightly. They can pack together very close to produce solids. So if you're eating something, like if you're looking at a piece of bacon, bacon is going to have, there's going to be some areas in bacon where there's a reddish brown where the meat is, but there are going to be some other areas where it's just a field of white. That means there are solid fats there. Those are solid fats. And so when you cook the bacon heats up, the fat changes from opaque to transparent, and you're thinking, well, and that's because you're dissolving the fat, or you're melting the fat to make a liquid. And then you eat the bacon, and while it's in your stomach, it's in a liquid form because they're not packing together. While they're in your intestines, they're in a liquid form. They're not packing together. But now when they cross your intestinal wall, go into your bloodstream, now they're going to start packing back together to become solids again. And then when they get to your coronary arteries in your heart, then they're going to start packing on the walls of your arteries, your coronary arteries, occluding them. So here, if we have here a coronary, let's say this is a coronary artery, and what's going to happen, those fats, the saturated fats, the ones that don't have a kink, they'll start depositing on the walls of your coronary arteries because they're very thin, and they're going to get very close to closing up. And then when you have this condition here where they're not all the way closed, you're not going to get enough oxygen to your cardiac tissue. That's going to cause chest pain, and we call that angina. So that condition is called angina. This is caused by saturated fats. And then one day, to keep on eating these saturated fats, that's going to close up completely. And now, let's say if we're going in that direction, any tissue downstream of this is not, any cardiac tissue downstream of this is not going to get any oxygen, and that cardiac tissue here is going to die. And so we call that a myocardial infarction. I-N-F-A-R-C-T-I-O-N. That is also known as a heart attack. Okay, and that's why straight or saturated fats are bad for you. But the bent ones, the ones that have the bend in them, the unsaturated fatty acids, they're very good for you because they don't pack together closely in your coronary arteries. Okay, let's give a little information about this one. We already know the name of it. Again, let's go ahead and go through it. The simple number name is going to be 16:1 fatty acid. The complex number name is going to be 16:1 delta 9, but it just has a cis orientation this time. This is the one that's found in us. We also have a systematic name. The systematic name is going to be I didn't give the right name before for the last one. It's going to be 9-hexadecanoic acid. 9-hexadecanoic acid. That's the systematic name. And the common name is going to be, just going to be palmitoleic acid. In a biochemistry course, you don't have to distinguish between, if something is cis, it's understood to always be cis. Only if it's trans do you have to indicate that. So if I just put palmitoleic acid, everyone in the world knows you're talking about trans, because we're talking about a biochemistry course or a biology course, because in biological organisms, fatty acids, the default is going to be cis. And so cis puts that kink and that makes these very good for us. So this is going to be an unsaturated fatty acid, but it only has one double bond. So it's unsaturated. It's unsaturated because it has a double bond, but because there's only one double bond, we can say it's mono unsaturated. And so at some point, probably not in this lecture, but at some point we're going to talk about polyunsaturated fatty acids that have a double bond in more than one location. And so this is all we're going to say about palmitoleic acid, a very common unsaturated fatty acids found in biological